Welcome, everybody. It's my pleasure to welcome you to the last session of what has been, for me, a truly remarkable day, and I hope it has for many of you as well. My name is Kate Smith. I'm a faculty member in Ecology and Evolutionary Biology, and it's been a real privilege and honor this year to serve as Interim Associate Dean of Biology for undergraduate education. So in the next 45 minutes, we have a series of 3 exciting TED-style talks from Brown Biology alumni and faculty. We've decided that we wanted to streamline this session, so our speakers won't be taking any questions, but certainly they'll be around at the reception that will follow, and we invite you to connect with them then. So without any delay, I'm very delighted to introduce our first speaker. Dr. Lucy Spelman received her bachelor's degree in biology at Brown in 1985. She completed a doctorate in veterinary medicine at the University of California, Davis, and she's a veterinarian board certified in zoological medicine. Previously, Dr. Spelman served as director of the Smithsonian Institution's National Zoo in Washington, D.C., and field manager for the Mountain Gorilla Veterinary Project in Rwanda. She now practices here in Rhode Island, and she teaches courses at the Rhode Island School of Design. Her research interest, similar to my own, is in the area of One Health, an emerging field that examines the interactions between animals, humans, and environmental health. So please help me to welcome Dr. Lucy Spelman to the stage. She'll be speaking with us. And her talk is titled Exploring the Art and Science of Saving Species. Thank you.
Thank you. Thank you, Kate. So I thought I'd start with this image that I took in Namibia about 10 years ago. And I'm pretty sure I took the photo because of the size of the elephant. I mean, that bull elephant just dwarfs the size of those zebra. I think I also liked how the ostrich were on either side of the elephant. So the male ostrich is the one that's black, and the female is the grayer feathers. I think I also was impressed. This was my first trip to a dry grassland in Africa. So in East Africa, you would see the same species, but it would be very green. Unfortunately, if I went back and to try to take the same photo today, I don't think we'd find that elephant. I don't think we'd find an elephant anywhere near his size. I think you all know that we have a huge problem with poaching of elephants for their ivory. We're actually sort of selecting for smaller and smaller elephants. So this is a massive elephant by today's standards. So the animal kingdom is shrinking. And in my career at the beginning, who would have thought that lions would be endangered? But they are. By the way, this photo, I did not see the lioness when I took the photo. I stepped out of the truck and took the photo. That is why they're colored that way. I was lucky. Whew. Okay, sad slide. So we are in a 6th mass extinction of species. My patients basically are going away. I was trained to treat all species, and the endangered species list is just getting longer. And sometimes I feel like nothing I do is going to help in the face of human impacts. So maybe I have a sick jaguar somewhere in this forest in South America. Even if I make him better, am I really going to help that species at the rate that we're mining this part of the world? So essentially what are called the extinction drivers, those are stronger than any medicine I have to treat an animal with. And this is something I started to think about in my career. Essentially, there's no treatment for an extinct species. Yes, we could maybe genetically engineer this giant river otter back, but why would we do that if its home is gone? So my profession is endangered, right? There's only 156 of us board certified in zoological medicine in the world. I started to think more, well, why is that? Because we know what to do. We know how to save species. This is a white tree frog, by the way. We have brought species back from the brink of extinction. This is a black-footed ferret, of which there were 17, maybe, left in the wild. This little guy is eating a black-tailed prairie dog, which is the only food of this species, which is one reason they went away. So we know how to bring a species back. We know that animals play all these key roles in society. Wild animals, domestic, urban. We rely on animals, and we know that. We also know One Health. We understand that our health is connected to animal health and to the health of the environment. 1 out of every 3 bites of food that we eat is made by a pollinator. Most pollinators are bees. We need healthy bees. We know this. We even know that we can save a species and make some money off of it. So this is a baby mountain gorilla, Endikazi, one of the cutest photos I have of any animal. Whole other story about her. But mountain gorilla ecotourism makes money to protect the gorillas. You go diving, the money you pay protects the corals. It does work. Our own species, though, is also in trouble, and we know this. This is an island off Panama. I was so struck by how many people were living in this tiny space, and there was no grass. There was not even any mud. Now, this is not good for humans. So we've had 5 prior mass extinctions, and the dominant species has gone away. So we are the dominant species, so think dinosaurs. This is a very happy illustration by a student of mine at RISD, but it's not a happy thought. We are in trouble too, and we know. So we know better, but we're losing species. And for me, the reason is that conservation works only if it's relevant to our daily lives. So if this topee antelope, if you never go to Uganda to see a topee antelope, you're probably not going to make a choice to help conserve it. Also, I think that we think of conservation as a science, and it's a whole lot more than a science. There's lots of different definitions. The one I use is it's a problem-solving process. It's so that humans can live in balance with our nature, both the resources we use and the biodiversity that we use. And that's not for the animals or for the place, it's for us. It's one of my favorite species, a giant anteater. So conservation, we have to all do it. And the decisions we make are partly based on science, but partly based on how you feel about the animal. So this is a pink-toed tarantula. Tarantula, how many? Spider. So our emotions around the animal have to do with whether we want to take steps to help it. And we need to understand those emotions and explore them. If you have no life experience with an animal, so this is my friend Kenneth, he's showing a bunch of tourists a parrot snake. Everybody's so excited in this photo. But lots of people, you say, snake? That's a negative. I think it's also about our sense of connection to nature, our willingness to feel compassion and feel for other people. So this little boy, he's holding an endangered species, a red-footed tortoise. But he's a Makushi Amerindian, and his culture is also endangered. So the two are connected. Meisheng and Chen, giant pandas, pretty easy to get excited about saving them. They're adorable, they're fun. I actually brought them from China to Washington. You should go see them. But we need more people saving more species than just these guys. So the way to do that is to make the problem of conservation and the solution more relevant to our daily lives. And this is a designed solution by one of my students at RISD, Chloe. So you care about the bees, get a planter. It comes with the seeds. The seeds sprout a flowering plant. The flower is food for the bees. So we can make conservation relevant, but we have to figure out what's happening to animals today and then make that meaningful and relevant. This is a watercolor done by a student who I took to Guyana who saw that giant river otter, and this was his interpretation. So my goal in teaching art and design students is I try to help them get the science behind the topic first and then use their art and design work in the studio to interpret it. So here is just a collage, and the topic was what's happening to giant pandas in the wild. So human development, road building is fragmenting the habitat, but the habitats are not that far away. And if we connect those forest habitats, we can help the panda. All of that's in this collage, even addressed a draft. So the potential of the students to get these ideas, make conservation relevant, has got me very excited. And so what I'm doing now in my career is teaching in the liberal arts division at RISD in what we call the science elective, Science for Art and Design Education elective group. And I teach these 4 courses that I've developed, and in all of my courses, students get to do a final project that combines the science with their art and design abilities. And some of the projects are a call to action, like this is like, okay, dengue fever is happening, we should be worried about it. Some of them are the solution. Okay, let's protect the Iberian ibex by doing this beautiful wine bottle label, because the last time you opened a bottle of wine, it was probably a screw top or a plastic cork, and the natural cork is the forest for this species. Some of them are just beautiful, like this EEV. This is a Hawaiian bird that's in trouble. It's a painting. This is a coral bleaching painting. So what I've been finding is that my students are exploring the artistic and humanistic side of what I teach them, which is the scientific side of what's happening. And I am encouraged by seeing that this collaboration can actually generate solutions. This is a papier-mâché jaguar, same idea, the forested fragmented. What can we do about it? How do we feel about it? So what I've done is launched a website called Creature Conserve, and the idea is to inspire more of this collaboration. It's basically a gallery of artworks by the students. You click on each piece, and you can get a little bit about the piece. Maybe there's a link to a science paper that they used. This watercolor is about overfishing. And then you can actually go to the artist's website. So I want to try to foster this collaboration that I am encouraged by. I think that we won't do any better at conservation unless we collaborate better. Maybe someday you'll see the bat stamps out there, you know, in the U.S. Postal Service. 1 in 5 mammals is a bat, by the way. We all need to do conservation. We all can do it. And I hope you'll take a look at the website, Creature Conserve, and I hope you'll think about all these animals that need all of our help. Thanks.
Thank you, Lucy. That was really great. So it's my pleasure to next introduce 2 of our more recent alumni, Joshua Cohen and Justin Klee. Joshua Cohen concentrated in biomedical engineering and received his bachelor's degree in 2014. He's the co-founder and CEO of Amylyx Pharmaceuticals, an early-stage biopharmaceutical company focused on neurodegenerative diseases. He's also the founder of Brown Biotechnology Investment Group, a student-run biotechnology investing group here at Brown. Prior to Amylyx, his research experience included work at the National Institute of Standards and Technology and here at Brown University. Justin Klee was a neuroscience concentrator at Brown and received his bachelor's degree in 2013. He is president and co-founder of Amylyx Pharmaceuticals. Justin has leveraged his extensive experience in neurophysiology and Alzheimer's disease research to help to build the company. So please help me in welcoming Josh and Justin to the stage. They will speak to us on the virtual biotech revolution, how 2 undergrads started a pharmaceutical company.
Hi, everyone. Thank you very much for that warm introduction. We're really excited to be here. So, I wanted to start with a quote from one of our heroes, Steve Jobs. So, on the founding of Apple, he said, "When I got started, I was 20 or 21, and my role models were the semiconductor guys like Robert Noyce and Andy Grove of Intel, and of course, Bill Hewlett and David Packard. They were out not so much to make money as to change the world and to build companies that could keep growing and changing." So with that inspiration, Steve Jobs and Steve Wozniak started Apple in Jobs' parents' garage in 1976 with a vision that they could build affordable, easy-to-use microcomputers and that it would be in the homes of everyone. It wasn't long after that before Bill Gates and Larry Ellison and Michael Dell and many other young entrepreneurs founded companies and started what became the personal computer revolution. Now, fast forward to the 21st century, and companies like Google, Amazon, Facebook, and many more have transformed the way that we interact with people and the way that we exchange merchandise and information. And it was founded by young founders on similar ideas that their predecessors had had. Now today, they also started what could be called an entrepreneurship revolution. Today, hundreds of thousands of young people flock to universities, to startup incubators, to startup competitions, to form what could become the next Facebook. And many of these have been very successful. Now, additionally, the whole idea that being young drives innovation has continued to be incredibly integral in this model. In fact, we found this recent study that found that of startups in high tech, the CEO age on average is just 31 years old. Now, there's been a similar revolution in biotech and biotech startups. Now, just to give a little brief background, I think when most people think about drug development, they think about the pharmaceutical giants, the Pfizers, the Mercks, the Novartis, Roche, et cetera. However, there's a very key place for small biotech companies, which are often spun out of universities. The reason for their existence is that they can take on projects that are more niche, maybe riskier, and develop those. If successful, they're often acquired by these large pharmaceutical companies and help them bring to market, or they IPO and become the next pharmaceutical giants themselves. Now, this model has been extremely successful, and just 3 brief examples are here. So the first is Vertex. Vertex was founded in the early 90s, now boasts about 1,800 employees, and has a radical cure, well, not cure, but radical therapy for cystic fibrosis. And has, for a disease that really had very few therapeutic options before, it's the first truly disease-modifying therapy. Pharmasets similarly developed a molecule which became Sovaldi, which has radically changed the standard of care for patients with hepatitis C, which is a similarly horrible disease which had very few treatment options before, and now is actually a cure for some patients. Mankind recently had FDA approval for inhalable insulin. So in the same way that you could inhale an asthma medication, now you can inhale insulin for diabetes, which again is just an enormous shift in what's possible. So I think all of these companies absolutely benefited from this startup biotech model and have changed the lives of millions of patients as a result. However, despite this potential, the rate of biotech formation and growth has remained about the same over the last 20 years or so. And I think we have to ask ourselves why that is. Why has tech been so incredibly successful with this model, but biotech has lagged behind? I think the first major issue is that traditionally, the barriers of entry have been way higher. So for example, if you want to start a biotech company, you have to have a lab, you have to have equipment, you have to have people to run the lab, you have to have regulatory expertise, you have to have medical expertise, the list goes on and on. It's a very expensive proposition. When you contrast that with Jobs and Wozniak, who started their company in Jobs' parents' garage, it's a very different proposition for investors as well as for founders. Now, contract research organizations have largely changed the game for that and allowed virtual companies such as ours to exist. Contract research organizations are companies that specialize in a particular field. So for example, we use Charles River Labs Discovery Services in Finland. They have a world-class central nervous system preclinical division over there. They have incredible researchers, most of whom are out of the University of Eastern Finland, which is a real powerhouse in neurological disease. They have expert facilities, and so we are able to design studies with them. They give us a quote, we pay for the study, they run it, the results come back to the company, and we analyze them. And in this way, Josh and I have been able to have a pharmaceutical company and run world-class studies without having our own brick-and-mortar operation.
So I think the other barrier for why these life science companies have started at such a slower rate could be summed up very well by what one of our potential investors once said to us, which was, kids don't do drug development. And looking at the statistics, this is actually pretty true. The average age of founding CEOs of life sciences companies have typically been in their 50s or 60s. There's kind of this cultural model that to be a biotech founder, you need to first get your PhD, maybe do a postdoc, work in academia or industry for many years, and sometime around when you're kind of a leader in the field, when you're in your 50s or 60s, then you're finally able to start a biotech company. And there's a lot of positives to this model. I mean, you get some incredibly experienced founders who really bring a lot of value to the company. But one of the issues with this is it makes an incredibly restrictive group of people who can actually bring new therapies to market for patients. And I guess to sum up too, innovation kind of happens in such a way that that it can happen at any time, any place. Long before somebody's in their 50s. To quote Steve Jobs, but innovation comes from people meeting up in the hallways or calling each other at 10:30 at night with a new idea or because they realize something that shoots holes in how we've been thinking about a problem. And this is actually exactly how Justin and I started this company. I'd been thinking deeply about Alzheimer's disease and had come up with a couple therapeutics, but they were still in a very early stage. And I ended up going to a friend's birthday party with Justin, and after probably a few too many drinks, we ended up talking in the hallway till about 4 a.m., and by the end of that said, look, we're going to found a company, this is a good idea, let's do this. And I think often that's exactly how innovation happens. A good idea, strikes somebody, and it's a problem if they have to be 50 or 60 and have to have completed just an incredible amount of work before they can actually make any difference with those ideas. And I think it's safe to say we've been fairly successful with this model. At this point, Amylyx is supported by a large team of advisors, consultants, board members, and investors, just some incredible people that we're lucky to have from both pharma and academia. We've completed multiple successful cellular and animal studies for Alzheimer's disease as well as ALS. We've been happy to get significant private investment. We're going to be meeting with the FDA for a pre-IND meeting in May to kind of finalize our plan to get to clinic. And we think with all this coming together, we should treat our first patients within a year or so. So it's been pretty successful. And I think that none of this would have been possible if we weren't able to contract more or less everything out. From our studies to our regulatory support, all of our legal support, we've contracted everything out, and it's enabled us to do this without the brick and mortar operation. And the other thing I think is really important is it's shown that this is possible. That kids can do biotech, and and I hope in a way that from what we've done, we pave the way for other kids or other younger people to do this kind of thing. And honestly, that's incredibly important too. You take a disease like Alzheimer's that we're trying to tackle, and Alzheimer's disease has not seen any progress in over 10 years. And one of the reasons for this is that lots of large pharmaceutical companies have just about completely cut their programs in Alzheimer's disease. And there's a reason they're doing this. There's been over 130 consecutive failures for drugs in Alzheimer's disease. And these bigger companies can't afford to take the risks, and have another kind of failure like that. So where the innovation is going to come in diseases like this is in these smaller companies. Companies that are able to take a little higher risk. Alzheimer's disease, has become just a global tragedy. There's over 5 million patients affected in the US alone. It's one of the most horrible things a family can go through. And actually, if left unchecked, it's calculated that it will crash Medicare within 20 years. So these solutions are needed, and they're going to come from these smaller companies, and we need lots of shots on goal. So I want to conclude today by talking a little bit about how Amylyx has been for us. For Justin and I think this has been one of the most incredible experiences of our lives. There's certainly been a lot of tough days, a lot of naysayers, a lot of people saying things like kids can't do drug development. But when the goal is kind of this large, it's easy to kind of keep pushing, to keep working, to try to make things happen. None of this would have been possible without being able to contract out just about everything. We hope that this model, similar to what we saw in the computing industry, where young people started innovating and all these amazing technologies burst forward, we hope this is going to happen in biotech and really change the standard of care for patients. Ultimately, I think it's safe to say Justin and I took pretty significant personal risks. We founded this our undergraduate year, instead of going to grad school, instead of going and working a kind of standard job. But I think it's safe to say that following your passions, doing these kind of things is what really makes a difference and what really makes a change. So I want to end off the talk by quoting Steve Jobs again. Your time is limited, so don't waste it living someone else's life. Don't be trapped by dogma, which is living with the results of other people's thinking. Don't let the noise of other people's opinions drown out your own inner voice. And most important, have the courage to follow your own heart and intuition. So thank you all very much. And I want to take a second before I go to thank Brown University. Gave us our education, made this possible, as well as Alan Harlem, Angus Kingan, and Alex Bronstein, who have helped us a lot at Brown. All of our consultants and collaborators at Charles River Labs, as well as our investors and board members and advisors. Thank you very much.
Thank you.
I think it's safe to say everybody in this room is totally proud of you guys. That was great. Thank you. So I'm pleased to introduce our final speaker for the session, Dr. Jeff Morgan. Dr. Morgan is a professor of medical science and engineering and co-director of the Center for Biomedical Engineering here at Brown. He completed a bachelor's degree at Syracuse University and a PhD at Harvard. He's had postdoctoral training at MIT and the Whitehead Institute. In addition to publishing more than 130 peer-reviewed papers, reviews, and chapters, he's edited 2 books and is an inventor of 12 U.S. and international patents. Dr. Morgan has received numerous awards and patents for his research in gene therapy and tissue engineering, and he's co-founded 3 companies, including his latest, Microtissues, Inc. Among his inventions are the 3D Petri dish, a technology for growing cells in 3 dimensions with applications in toxicity testing, drug discovery, and tissue engineering. Time magazine named the bioartificial ovary developed using the 3D Petri dish as one of its top 10 medical breakthroughs of 2010. So please help me in welcoming Dr. Morgan, who will ask the question, can we build organs?
Great. Thanks, Kate. I have to say, Brown students are absolutely awesome. The work I'm going to show you is the work done by Brown students. So let me cut to the chase and lower your expectations. The answer to this question, can we build organs, is no. Take care of your own organs now. However, there's cause for excitement, and that's what I'd like to expound upon in this 12-minute talk. We are in what I call the age of diffusion. This field of tissue engineering has been working in a way because it relies on diffusion of nutrients to it, and that means it's limited by size. We're about to move into an age of profusion, and I think then, and only then, can we begin to think about possibly building organs such as the kidney, liver, heart, and pancreas. So what I'm going to do is talk to you about some of the advancements and some questions that make a lot of sense to me. Some are a little offbeat, some are on mark. But we do know the problem. The medical problem is severe. The shortage of organs is just getting worse. And because we're here at Brown, we're very socially responsible. So we know, in fact, that there's a sad, ugly black market that exploits people in poverty in the international community. Now, those are all serious problems, but there are some key questions that we need to understand. And the first question is, well, will I win a prize if I build an organ? Seems a bit of a strange question, but the answer is yes. Turns out the Methuselah Foundation will provide $1 million to the first group that will bioengineer a liver solution for a large animal, keep it alive for 90 days. This is the era of big science, prizes, XPrize. This is for real. Now, that's intriguing, but aren't companies actually building organs? Well, to some degree, yes. Oops. To some degree, yes. There's a company called Organogenesis about 30 miles from here that's been growing skin, one of the first companies in tissue engineering, and they commercialize it, they sell it. The new kid on the block is a company called Organovo, which is using bioprinting to try to build organs. But they have a ways to go. But here's another interesting question. Well, okay, if you want to build an organ, how many cells are there in the body? It turns out there's a great little paper that has answered this question for the 70-kilogram man. The answer is, in fact, 37 trillion cells. And here's the breakdown. We're mostly blood. I hate to disappoint the neuroscientists, but we're mostly blood. And in fact, we're wrapped in skin. If you go to the major other organs, you can see that here we are at liver, kidney, heart, and pancreas. So these are serious numbers, but they're actually finite numbers. And they're numbers that I will try to impress on you are possibly achievable given some recent breakthroughs. So that's encouraging. We have a finite number of cells. But let's do the thought experiment. If I have one cell and it doubles every 24 hours, how long will it take me to have enough cells? Well, here's that mathematics. Number of cells versus days. There's a pancreas, there's a heart, there's a kidney, there's a liver, there's skin, there's human. So we've already engineered skin. They do it quite well. But these 4 other ones are under a lot of investigation at this point in time. But that's just theory. That's just mathematics. Does it match anything that makes any sense with respect to the need in organs? Well, here's the wait list. Here's the time to wait. So that's a reasonable time to build an organ and supply it to a patient. How does that stand up to the old-fashioned way of making organs? Well, there's my new granddaughter. Beautiful. She did it in 266 days. But the design constraints are extraordinary, right? They're all packaged. All the organs are done at once. What we learned from skin is there's no need to do that. They can grow acres and acres of skin because they've released it from these design constraints. But there's our theory. There's our current model. Now, we all know about the amazing breakthroughs in iPS cells. These are induced pluripotent stem cells. Take an adult cell, reprogram it, make it primitive, and then it can differentiate into all the cell types. Extraordinary breakthrough. It's moving at lightning speed. Discovered in 2007, Nobel Prize in 2012, clinical trials in 2014 for degenerative eye disease. The pace of innovation, as you heard from our earlier speakers, is accelerating, and that's incredibly exciting. Here's another paper. The Schaefer lab at Berkeley. They grew iPS cells and asked the question, how long can we expand them in culture? These are extraordinary numbers. Let's put them back on the graph that I had, the theory graph. In fact, here they are. A 20-fold expansion every 4 days, and they took them out to extraordinary numbers. So the answer here is really we have the raw materials. We have the cells, the most primitive cells that can differentiate. We can grow them at scale. We can make lots of them. And many labs are working on ways to differentiate them down all these various lineages. OK, that's encouraging. But what are we doing here at Brown? So with that notion of you've got the raw materials, but the individual cells, we now need to think about ways to assemble them into larger structures. And our approach is to start with building blocks. And my lab, several years ago, innovated and came up with this little device, this catchy name of 3D Petri dish. It's a very simple device. It's a mold that we put a gel in. We pipette cells. And then the cells will actually self-assemble into little spheroids. And here's the process. Remove the mold, fill it with cells. They settle by gravity, and then they self-assemble. Here's a time-lapse video of that process. Hundreds of these are forming, aggregating. The cells are self-assembling in each of these small molds. And of course, as mold designers, we have control over that, which I'll show you some interesting data. So, very simple process. You just make the mold, you add cells to that. Now, is it the same density as a tissue in an organ? In fact, the answer is yes. From this paper, we can calculate the cell density of the liver. Here's our spheroid building part. And in fact, the density is comparable to that of the liver. You can see it right here in the H&E section of liver, H&E section of one of our little spheroids. OK, so we've got these interesting building blocks. And we can make lots of them. Here's a 24-well plate. Well, we can make more of those plates. We can scale this up fairly easy. Maybe we can even automate it. That's interesting. What kind of biology is happening? Well, it actually turns out to be fairly complex. So in this time lapse, we have 2 cell types that have been mixed, labeled. They're self-assembling. They're self-organizing. That's an interesting property. The mold system works with a variety of cell types. These are cardiomyocytes, cardiofibroblasts that are self-assembling and self-sorting. The cells will differentiate in 3D. Here's a confocal stack of epithelial cells, breast epithelial cells, that after they've self-assembled, they form these open luminal structures, these lobular-like structures. And they're doing this all on their own because they're pre-programmed to some degree to self-organize. Here's a series of spheroids of increasing size, and they've actually formed a nice little capillary network in response to the hypoxia of a large spheroid. So there's a biology here that's incredible that plays out all on its own And all we need to do is begin to understand some of these processes. But OK, we can make a spheroid. Why don't we just make a really big spheroid? That's what engineers would like to do sometimes. But the answer is no. And the answer is no because of this diffusion limit. And the larger you get, you have a limitation with respect to nutrient supply in and waste products out, and you get a necrotic core. OK, that's a bit problematic. If all we can make are spheroids, we can't build anything. Well, let's go back to our mold system. We have this 200 microns, and that's roughly the distance that you can make one of these tissues and not worry too much about diffusion. 200 microns, how about we make a rod-like structure? In fact, we have. Given the shape of the mold, the cells will self-assemble a rod-like structure. That's interesting. How about a donut? Same type of deal. 200 microns thick. We can have a dimension such as that, diffusion through the center. Here's our mold system. Here are some of our donuts. Now, we can also control this lumen size. As long as we keep this thickness 200 microns, we now are making building parts with a little more complexity, a little more size. A heck of a lot more cells in a spheroid. Let's try to make something a little more complex. In this case, we made a honeycomb structure, a series of donuts that are interconnected. Here's the mold. A very talented undergraduate in my lab made this very large honeycomb, which is approximately 2 centimeters across, 6 million cells. And it's an extraordinary structure because it's self-assembled in 24 hours. So we have these interesting building parts. They're significant in size. We've gone from a simple spheroid, which was about 1,000 cells, to a small honeycomb, which is 250,000, about 250-fold increase. This large honeycomb, 6 million cells, another 24-fold increase, equal to about 6,000 spheroids. So we have these interesting building parts, but can we build with them? And the answer is perhaps. These living building parts were fused. So what we've shown in the lab is if you bring parts together, allow them to touch, because they're living, they'll actually self-fuse, sort of a biofusion. Here's 2 donuts on their edges, 2 donuts stacked. Here's a pile of donuts that are interacting. But that seems really quite random and not easy to control. So as bioengineers, we dreamed up this device. Inspired by the electronics industry that uses vacuums to pick up parts and assemble circuit boards, we came up with this device, the Bio Pick and Place. Here's how it works. A pump is pulling fluid through this gripper head. Comes down, picks up this living building part, moves over here, stacks it on another building part, and then flows fluid through this. We're perfusing. Just as the heart perfuses all the major organs. And that's why it defies diffusion. That's why we can have tissues and organs that are large in size. Here's a couple scenarios for various kind of building strategies, open structures with large numbers of cells. Now, we're not at the point where we've made organs, as I said at the beginning, but we've got some interesting results with that. Here's version 1.0. This is what you can get with $200 at Home Depot. Okay. Here's the gripper head, plastic box. It's in media. The gripper head has a membrane here. It's a small pores, about 3 microns. Suck fluid through that, and you can grip and move parts. Andrew Blakely was an MD fellow at my lab. Very carefully stacked 8 toroids, 16 toroids on a capillary tube, 170 microns outer diameter. Placed them. You can see over time, they're actually merging and they're forming a tube-like structure. Pretty interesting. We went on to doing the pick and place with the honeycomb structures. Here's our holding pen. Pick up the part, bring it over here, stack them, try our best to align them. In the meantime, we're perfusing. This is a motion. This is in motion, this whole thing. And we're sucking fluid through this stack of tissue. Here's a stack of them from a view from the side. So, Home Depot, $200, a couple of pumps for my chairman. Thanks, Wayne, if you're out there. Let me summarize with this slide. I think we have the raw materials, these IPS cells. We can grow them at scale. We have building blocks. They self-assemble in 24 hours. We can fuse those building blocks in 48 hours, so we're starting to get a reasonable time for assembly. We have cell densities that approximate organs, 2 or more cell types. There's this biology of self-organization and differentiation. We need to learn more about that, how to control that. They will form capillary networks. The lumens that we have in our stacks are still too big. We still need to understand how to make capillary beds in these building parts and connect them all up in a perfused-like system. And here's the scale. Spheroid, 1,000 cells, up to our largest so far, 6 million. We're going to push the envelope on this and see how large of a part we can make. But back to the beginning. How many parts? 6 million cells in one part. To make a liver, 50,000 parts. Kidney, 1,600. Heart, 1,000. 500 parts for a pancreas. Now, I don't want to underestimate the complexity of these organs. They really are quite complex. But I'm excited because I think we now have a finite number of parts. They seem reasonable. They seem like this is an area where we should continue to push. And we're very excited because of some recent funding that we got. We just got an award from the National Science Foundation for $1.4 million to build version maybe 3.0. And so we go from $200 at Home Depot, about a 7,000-fold increase in our funding. We already have joystick control. We already are thinking about vision recognition and automation. And I'm excited because we're working in a realm where we can bring in all of the stuff that's understand about automation and bring it into this space where we're taking the raw materials of cells and trying to assemble them and differentiate them into organs or proto-organs that might be useful for disease. All this work would not be possible without a lot of very talented Brown students, Tony and Dylan, early guys with the molds, the latest, Kaylee, a master's student, Andrew Blakely, an MD fellow, really pioneered the biopick and place instrument. And we're on a verge of, I think, a very exciting chapter. Still a lot of work to go, as I said, at the cocktail hour. Take care of your own organs. Thanks for your attention.
Well, thank you. Please join me in thanking these speakers for 3 very inspiring talks.